Well, look, thank you for that wonderful introduction, and thank you for coming. Can you hear me? Is that loud enough? Yeah, that's good. It's a real pleasure to be here. It's always a pleasure to come to Vienna, but also particularly to IMP and to this complex, which is a real jewel for molecular cellular developmental biology in Europe. And it's an honour to give a Birnstiel lecture. I knew him when I was in Edinburgh, when I was a postdoc in my 20s. He's in founding IMP, and the complex round here has caused a transformation, I would say, in molecular biology in Austria. So it's a real honour to be here. I'm going to talk about cyclin-dependent kinases. I'll let you turn on. I'm hopeless at computers, so I need assistance for one thing. I'm going to talk about what I'm best known for, which is cyclin-dependent kinases and the control of the cell cycle. But what I'm going to argue is perhaps contrary to the general paradigm that is thought to apply to the cell cycle, which is, and I shall explain in a little more detail, that a range of different CDKs arise and disappear as you proceed through the cycle, phosphorylating different substrates to bring about different events of the cell cycle. I'm going to argue that at least in fission yeast, this can be brought about by a single cyclin-dependent kinase, which makes us think more about the level of activity rather than qualitatively different protein kinases. And I shall use a combination of genetics and phosphoproteomics to investigate this particular problem. I started this particular work when I was at Rockefeller. I've continued it in the Crick. And I'm going to start by showing you the Francis Crick Institute logo, and I'm going to be assisted here because this is actually the most, if we can get it to work, the most interesting slide in the entire talk. Okay? So if you can activate it, and it's a little movement, then we can all be excited, and then some of you can go away if you're not. Look at it, a dynamic logo, the first you have ever seen. This is the Francis Crick Institute. It looks like this. It is next to the Eurostar terminal in King's Cross and St Pancras, and we take the building over in June, May or June, and I'll move the three institutes that are merging into it over the rest of the year. So it will be about 1,400 scientists, which I think are the numbers which are on this particular complex. It is, I have to tell you, a total logistical nightmare, and a total financial nightmare, and a total institutional nightmare. But we won't talk about that anymore, okay? It is, however, there, which is wonderful. Right, I'm going to give you an introduction and run through some of the early work a little quickly, because it has been published several years ago, but you need it just for the introduction. So, good. This is a figure from David Morgan, very good book on the cell cycle actually, which tells you the canonical view of how the cell cycle is regulated. Just as I said a minute or two ago, different CDKs, which are enzymes made up of a catalytic subunit and a cyclin to activate it, appear and disappear as you proceed through the cycle. They bring about progression through G1, they bring about progression into S phase. In G2, they block the initiation of S phase, and then, as it was discovered, at the G2 to M transition, they activate mitosis and then start the cycle again. And the concept here, to which many people contributed, including my own laboratory, is that qualitatively different CDK activities have qualitatively different outputs on phosphorylating a range of different substrates to bring about that wide variety of different functions, because it's a very complex set of processes of DNA replication and mitosis that have to be initiated there. And I'm sure what I've just described is probably what most of you were taught as undergraduates. Am I right? Please nod, otherwise I shall feel depressed. Okay? Yeah, more or less right. Okay. And it's subject to a lot of regulation. We'll touch on some of it. There's the cyclin CDK interaction. There's a negative phosphorylation on tyrosine 15, bottom right, an activating one on a threonine, 167 in fission yeast, 161 elsewhere. The CDK inhibitors, a whole complex network. You just have to be impressed. You don't have to remember any of this, okay? If you look around the Cyclin CDK, you'll see that it's regulated in a wide variety of different sorts of ways, many of which have been studied in great detail. And what you should take away from it is that it is actually quite complicated, okay? It's quite complicated. And if you then try and model it, as people tend to do, then you end up with something looking like this. Now, you can't read that, can you? And I don't want you to read it. I only want you to become depressed looking at it. Because there is a problem we have in modern biology, and I'm going to address this a little bit at the end, is that we think simply by describing and noting what is involved, we yield understanding of what is happening. And although describing and knowing what is involved is absolutely necessary, it does not automatically lead to understanding, and indeed can obscure understanding. And quite a large part of my talk is trying to tease out of the complexity core principles and understanding of what's actually happening. And at the end, I'll say a little bit more about that. But that's the general approach that I'm going to try and describe. Now, for some years, I've been worried about this sort of stuff. Not because it wasn't necessary, but because it was not yielding really good understanding. So I tried to persuade postdocs that came to my lab, or graduate students, that perhaps we could try and simplify the network that would then allow us to focus on what were core principles. And we understand what the network is and we try and simplify it. A postdoc came to my lab, now runs his own lab, Damien Cadrusa in Rennes, in France. He likes RISC, that's him hanging off the end of this thing, at least he tells me it's him hanging off this thing. And so he decided to tackle the project of trying to simplify the cell cycle control network to identify principles. Introduction, fission yeast, simple organism. We have a range of CDKs, but only one catalytic subunit, CDC2, the catalytic kinase of CDK1, 4 cyclins, CDC13, SIG1, SIG2, PUC1, which are involved at various stages as we go through from G1, SG2, and mitosis. So fairly similar to the canonical view that you would have from studies in all eukaryotic organisms. So how do we simplify it? Well, the first thing that we decided to do was actually very simple, and I was surprised it had never been done before, which was simply to make a chimeric protein between the catalytic CDC2, catalytic subunit, and the cyclin activating subunit. Because what that does is remove all the functions that are needed to put those proteins together and to make a stable complex, because it's a single chimeric protein. So the first thing that Damien did was to fuse CDC13, which was the major G2 cyclin, to CDC2, played around with different linkers, put it behind the CDC13 promoter, and so on, to see whether we could get an active CDK with the intention of trying to eliminate some of the things here, like complex formation, that would no longer be necessary in this construct. And that worked. He made a fusion protein, which of course, this is a Western, this is wild type, this is blotted with cyclin antibodies. The chimera shifts it up the gel, of course, and we lose the original CDC13. This is what wild type fission yeast looks like. This is a septum dividing the cell in two. As you heard, it grows simply by cell elongation. Here we have a construct where we have the linked chimera, CDC13 linker CDC2, inserted into the CDC13 site, deleted the endogenous CDC13, deleted CDC2, and you can see the cells are perfectly viable. Okay? So it's being driven by the chimeric protein. Encouraged by that, Damien then marched through the genome and deleted SIG1, SIG2, and PUC1, which means that we now have no other cyclins in this strain, no other CDKs in this strain, except the chimeric protein. These cells look perfectly normal. They grow perfectly normally. If we look at the generation time, it's essentially unchanged. Even if we look at rather more subtle parameters of cell cycle progression like size at division, it's more or less the same. And if we look at FACS analysis, it's the same. Just so you get the nomenclature, this is wild type, this is the linker, this is deleted for CDC2 and CDC13, and CCP is deleted for SIG1, SIG2, and PUC1. So what you appreciate, of course, is what this means is that, taken at face value, this cell cycle is being driven, as far as we can tell perfectly normally, by a single cyclin-dependent kinase, and we've eliminated all the other qualitatively different CDKs. This is not completely unexpected because what is termed sort of plasticity in metazoan cells of eliminating different CDKs has come up with surprising results of what people call plasticity or redundancy that has been slightly swept under the carpet because nobody quite knew what it meant. I'll refer back to that a little bit later. This strain behaves reasonably normally if we tag the fusion with YFP. Then we can order the cells in the cell cycle according to their cell length, as you can see here. What you see is what happens normally, which is that the chimeric protein accumulates in the cell cycle in level, primarily in the nucleus, and as the cells go through mitosis, it's disappeared because it's broken down by cyclin degradation, as shown by my colleague Tim Hunt, now many years ago. Now, because it was a little out of the box to suggest that the cell cycle could be driven by a single CDK, Damien and myself decided that we should be as critical as we could in establishing that this was really what was happening. So we constructed a strain where we could switch off either the CDC13 moiety or the CDC2 moiety, either by introducing a TS mutation there, which we had identified many years before, or introducing a shocker mutation within the ATP binding site. I turn poor old shocker into a verb and say we shockatized the protein, because what essentially that does is widen the ATP binding loop, so a bulky ATP analog can get in there and inhibit this kinase, but at the right levels of the analog, no other kinase in the cell. So we can specifically inhibit just the CDC2 kinase activity. This is this experiment. What we have here are cells and fission cells and stain for nuclei. This is at 25 degrees when this part is active in with DMSO, so no inhibitor, the cells just grow normally. If we add the SHOCAT inhibitor, what you can see is within a generation, the cells cannot undergo cell division. They can continue to grow because they're not defective in RNA or protein synthesis. They elongate and grow, and they're uninucleate. They are unable to complete the cell cycle. If we keep it, don't add the inhibitor but shift to high temperature, we get the same result. So inactivating either this component or this component completely inhibits cell cycle progression. Further analysis, which I won't show you, shows actually it's blocked at two stages in the cell cycle. It's blocked both at entry into S phase and entry into mitosis. So it's required for both of those components. So this, we believe, was good evidence for saying that a single CDK can drive, at least in this simple eukaryote, the entire cell cycle. I'll return to that in a moment. But there is another sort of cell cycle, which is the meiotic cell cycle. Doesn't receive the same attention as the mitotic cell cycle, but we can study meiosis very straightforwardly in fission yeast as well. And I'll just quickly show you the experiments we've done in meiosis. This was done by Pilar here from Spain, who was a postdoc in my lab, just left a few months ago. This is the meiotic cell cycle, and what I want to just show you is that we have the single CDC2. We have CDC13, SIG2, SIG1, and PUC1, but two further meiotic specific cyclins, REM1 and CRS1. So there's now 6 cyclins. So now what we did was a series of experiments, well, you can imagine it already, where we deleted all of the cyclins to see whether we can drive the entire meiotic cell cycle by a single CDK as well. This didn't initially go immediately straightforwardly, because when we introduced into this strain here, which is in fact not deleted for for the two meiotic cyclins, and asked whether the fusion protein could drive meiosis, what we found was it could get to the second meiotic nuclear division, but was then arrested. So it couldn't rescue all of meiosis. we reasoned, having done Western blots and so on, that maybe the level of expression of protein was important. So we did a dead simple experiment. We just increased the gene number of the fusion protein. We put it up to 4, and then found we could get very efficient meiosis in the strain deleted for the 4 mitotic cyclins. Then we deleted, and now we go to CCPRC, so we've now deleted all the cyclins, and we get pretty efficient meiosis. It's going up to nearly 90%, whereas here in the control it might be 95%. So essentially, the single fusion protein, if expressed at the right level, will rescue not only the mitotic cycles, but also the meiotic cycle as well. So a single CDK can do all of these complex and different processes in cell cycle progression. We were a bit surprised by this, I have to tell you, having spent about 25 years of my life showing how complicated it was. Showing it could all be done by one single CDK was a bit of a humbling moment, I have to say. But it's good for you after you reach a certain age to be humble. Now, how can we explain that a single cyclin-dependent kinase can bring about all these different sorts of events? And let's just think about the mitotic cycle. It can bring about progression through G1, preparation for S phase, initiation of S phase, subsequently blocking initiation of S phase, which is a function less talked about, but also in G2, and then mitosis, completely different set of functions. This is a very complex series of outcomes to be brought about by a single cyclin-dependent kinase. The only model that we could come up with is shown here. It's one that we had played with some years before, so it wasn't completely new for us, which was a very simple one, which is that the primary driver of progression through the cell cycle is in fact very simple. It is a gradual rise in CDK activity, or at least a continuous rise, maybe not always gradual, which at low levels of CDK activity bring about one set of events, and at increasing levels a series of other sorts of events. And that's sort of reasoned, that's shown here. So we go from a low level at the beginning of the cycle, we bring about S phase, we then block S phase, at a high level we bring about mitosis, then cyclin degradation sets us back to 0. It could not be a simpler model. I mean, completely different to the complex network that we described. Now, we're now in a position to test that more specifically, as Damien did, because using the shockatized fusion protein, we can adjust in vivo the level of CDK activity by adding different levels of inhibitor outside the cell, which will adjust the level of CDK activity within the cell. And what that would mean is if you only need a low level of activity to get into S phase, you will need a lot of inhibitor to block S phase. If you need a high level, you will need only a small amount of inhibitor to block mitosis. And interestingly, if this model is as it's said here, if you're in G2, you can be directed to go into mitosis or into S phase simply by manipulating the level of CDK activity, because by dropping it low and then letting it rise again, you'll undergo S phase. So there'll be no arrow of time in the cell cycle. It's driven by the level of CDK activity. So Damien went on to test that. First of all, G2 to M, synchronized cells in G2, released them in varying levels of the shock-at inhibitor. Without inhibitor, you get a peak in mitosis about here. As you add increasing levels between 25 and 300 nanomolar, you delay the onset of mitosis until by 300 nanomolar you block it altogether. So all you have to remember from this experiment is that somewhere between 0.1 and 0.3 micromolar of inhibitor is what is required to block onset of mitosis. Just remember 0.3 micromolar, okay? I know it's difficult to keep numbers and things in your head. Just remember 0.3. Now you can go to S phase. S phase, and each of these points is a different FACS profile looking at 1C, 2C, or 4C levels of DNA content. And if you synchronize cells as they proceed through S phase, what you can see here is because G1 is very, very short, without any inhibitor at all, you see no accumulation in G1. If we now add the shock-at inhibitor, we see a little peak of G1 cells appearing because they're being delayed from going into S phase. And now you can see if we add increasing levels, and we went up to 10, you increasingly delay onset of S phase. So G1 is extended, and as you can see here, by 90 minutes. If we added 10, it would be extended by 150 minutes. So now what you can see is to actually delay and block onset of S phase requires 5 to 10 micromolar of inhibitor, whilst to delay and block mitosis is 0.1 to 0.3. So that's consistent with the model. Now, what about the most interesting one, which is, can we take a G2 cell and make it undergo mitosis or S phase simply by modulating CDK activity? And if we block cells in G2 with a moderate level of inhibitor, wash the inhibitor out, you go into mitosis. That's an obvious experiment, but that's the control. The more interesting one is to block it with moderate level of inhibitor, 1 micromolar of inhibitor, then give it a pulse of 10 micromolar, dropping the CDK activity to a low level, and then letting CDK activity rise. Now what happens is, if that happens, you see the cells undergo re-replication from G2 and actually become, of course, a higher ploidy. So you're programming whether a cell goes into S phase or mitosis simply by playing around with CDK activity. So just to roll out the cell cycle again, you've seen this already, we only need a small amount of inhibitor to stop this, consistent with this, a large amount to block this, and if you are here and let CDK activity rise, you go into mitosis. If you are here and force it down with 10 micromolar and then let it go up, then you will enter into S phase. Now, how is then all of this actually brought about? Well, to do this, we had to go to phosphoproteomics, because up until now this has all been, of course, genetics and physiology. And by the way, I think physiology is really important for studies like this. Often it's sort of not done as well as it should be. Often the biochemistry is beautiful, but it's not based, if it's not based on good physiology, then you don't quite understand what's happening at the level of the cell. So we now have a phenomenon, and the phenomenon which we're going to try and investigate is how do varying levels of CDK activity bring about these very different events. So this was done by a graduate student in my lab who's still with me, Matthew Swaffer, with tremendous support from our mass spec facility run by Brams Snyders and 2 technicians within that facility. And I have to say, what I know about mass spec can be written on the back of a very small postage stamp. Okay, so the success of this project is entirely dependent upon Matthew, with great support from our facility. That's just to tell you not to ask me a difficult question about mass spec, because you'll only get a stupid answer. Now, of course, how can we order a single CDK order events? We need to know about CDK substrates. We need to investigate that. We can use, of course, phosphoproteomics. So the first point is, what are the substrates for CDKs in the cell cycle? Now, strangely, you'd think we'd know, given that CDC2 was discovered in 1975, you'd think we'd know more about it than we actually do. And in fact, the studies have been rather limited. People have looked for consensus sites and done some phosphoproteomics through the cell cycle, particularly in human cells, but that doesn't tell you whether it's directly phosphorylated by CDK or just dependent upon it. So what we did was an extensive phosphoproteomics analysis to identify what proteins become phosphorylated in the cell cycle. We found about 5,000 sites that we've analysed, but I'm just going to focus on the CDK consensus sites. And then what we did is inactivated CDK activity just by adding the inhibitor, and only looked at those sites which rapidly lost phosphorylation. And here you'll see some of them here. This is a particularly nice one, where 4 sites in cut 3 become almost completely dephosphorylated within minutes of adding the inhibitor. Sometimes it's a little slower, as you can see here. Sometimes it goes from a lower level. But we identified a collection of around 250 sites which are defined by a strong consensus site and which are inactivated very significantly within about 15 minutes of inhibiting the enzyme. So we have now a library of good CDK sites. We don't claim it's every site, because there will be some that will be more stable, but I think we can claim that this is probably the best, most authenticated, if you like, collection of phosphosites through the cell cycle. We identified 250 approximately that came up in mitosis, and interestingly, only around 30 that came up around S phase. So there's many more phosphorylation sites occurring at mitosis than there are at S phase dependent upon CDK, at least in fission yeast. This is the half-life, which I'll come to a little bit later. Now, what we can now do is look at those events through the cell cycle, which we've synchronized here. I don't need to go through it. Basically, you have 2 rounds of cell cycle. We've synchronized the cells, and we've taken phosphoproteomic samples throughout these cell cycles. This is a lot of time on the mass spec, it has to be said. And we've looked at mitotic substrates and S-phase substrates as you go through the cell cycle. And this is the first thing that I want to draw your attention to. Let's look at the mitotic substrates. These cells are arrested in G2. They're released into mitosis, and just as you'd expect, you get a massive increase of these. You then go down to a low level, and then the second less synchronous culture, it comes up again and goes down. Each one of these is, of course, a phosphoproteomic assay. If we now look at the S-phase substrates, there's over 200 in here, and these are all just averaged. If we look and we look at 26 in this case, because we have to see them at every time point. You don't always see them at every time point, so we've been quite critical here. The first surprise we had was this. In G2, the S-phase substrates were still highly phosphorylated. They hadn't been dephosphorylated, which is, in fact, what I thought would probably be the case. You then went through mitosis, and they all underwent dephosphorylation, and then rose almost immediately, because cells undergo S-phase about here, to undergo S-phase, and then continued to rise as cells proceeded through G2 into mitosis. They never became dephosphorylated during the cycle. So the paradigm that it goes up and down in the cycles is simply not correct. It's simply what we've assumed to be the case. You get phosphorylation of S-phase substrates early and not of mitotic substrates, but the phosphorylation never goes away. So that's the first, sort of, if you like, correction or change or altered view as to how these sort of work. Now, I'm talking a bit quickly, so this is just to show you that it's built on data. This is the data that went into it on individual proteins. That's just a repeat of that. But I haven't got the time to talk about it. Now, given that, why do we phosphorylate proteins required for S-phase early in the cycle and not mitotic substrates? What can be the reason? Well, the obvious explanation would be that you can phosphorylate S-phase substrates with a lower level of CDK activity. And if that is the case, then we would get information by looking to see what level of inhibitor, of CDK inhibitor, is needed to block the phosphorylation of particular substrates. Are you with me? So this is what this experiment is about. And here what we see are a variety of mitotic substrates, and this is specific proteins. And what we've simply done is asked what level of phosphorylation is there at these sites with different levels of shock-ACT inhibitor. And what you can see here is mitotic substrates, these ones here, I'll show you an average in a moment, a sort of 50% reduction is occurring somewhere around 20 to 100 nanomolar in both of these cases. Okay? If we now look at S-phase substrates, we get a completely different picture. You require between 1,000 and 3,000 nanomolar of inhibitor to block phosphorylation of those substrates. So our conclusion is, and I think that should be on average on the next slide here, this is where we've averaged 147 mitotic substrates, 15 only of S-phase substrates here, because again we need a positive signal for every time point. And if you now look at this, what you can see is exactly what I just told you. The average here is around 100 nanomolar, the average here between 1,000 and 2,000 nanomolar. So the simple hypothesis is that M-phase and S-phase substrates are differentially sensitive to different CDK levels, and we bring about phosphorylation of S-phase substrates early because you only need a low level of activity. And that low level of activity is sufficient to phosphorylate S-phase substrates efficiently to bring about S-phase, and you require a higher level to bring about mitosis. Again, could not be a simpler model of how the cell cycle is regulated. Now, we can go further. And I want to tell you about this experiment because I think it is actually potentially interesting for those interested in protein kinases. Because what this allows us to address is in vivo CDK activity, which has been rather something that's been difficult to do over the years. You can use different reporters, for example, that look at specific kinase sites, but tend to be a little bit artificial. But we can use phosphoproteomics in a very simple way. We can take a cell which is phosphorylated on its substrates. We can add inhibitor, so we dephosphorylate in vivo the substrates. Then we can add back kinase activity by washing the inhibitor out, and then look at the rate at which different phosphosites are phosphorylated in time when they're going from essentially zero phosphorylation or very low phosphorylation, and get an instantaneous rate in vivo of CDK activity, and which is giving us an idea of what the in vivo CDK activity is. All of these things are prone to, of course, caveats, but it's certainly more direct than breaking cells opening and measuring CDK activity, which of course the traditional approach. And what you see here is that looking at phosphorylation of early substrates, that the level of activity that you see in G1 cells in this sort of experiment in the first 10 minutes of adding back is shown by these different levels in G1, S phase, G2 and mitosis. And what this tells us is in vivo CDK activity is increasing as you go through the cycle. This is an in vivo measurement of CDK activity. Of course, in vitro activities largely reflect this, but there hasn't been attention paid on the difference quantitatively in CDK activity at G1S compared with G2 to M. And this increases about 30 to 40-fold throughout the cell cycle. So that's, if you like, the dynamic range that we have to convert the analog behavior of CDK activity into the digital readout of G1 S phase, G2, and mitosis. We can now, in addition to that, and by the way, we were looking there at 10 minutes, but we've actually used the first 2 minutes to get a better sense of what that activity is. So we look at instantaneous phosphorylation. So that's the rate we look at. But we can now also look, again in vivo, at how different substrates respond to these different CDK activities. And you can see that shown up here. This is also looking at the first 2 minutes. And what you see is that late substrates hardly get phosphorylated, whereas early substrates, G1S, get very efficiently phosphorylated within half to 1 minute, showing that, again, in vivo, that the early substrates acting at G1 to S are much more efficiently phosphorylated by these very low activities compared with late substrates, which require these very high activities, which appear later in the cycle to bring about this phosphorylation. So this then translates, if you like, that physiology into a simple biochemistry. CDK activity associated with a single CDK is gradually increasing, not gradually, continuously increasing. There will be a different pattern as you proceed through the cycle, and you phosphorylate early on S phase substrates, and later substrates get phosphorylated later to bring about entry into S phase. You can also reorder the cell cycle. I won't talk about this because I can see some blank faces beginning to appear with all this damn data. We can reorder the cell cycle. I'll just say this, and those who are sort of still following it closely can look at the data and get it. Because remember, we can take a G2 cell and we can make it G1 and go into S phase. We can look and see what substrates are phosphorylated. It all works. In other words, if we kill CDK activity and let it come up again, we phosphorylate S phase substrates first, and that's why we get S phase. That's what was on that slide. Now, another factor that I briefly mentioned as we went through, but I now want to emphasize a bit more, is shown here. Is there an inherent difference in phosphorylation turnover on substrates that are phosphorylated early versus late? I actually thought turnover for late substrates may be much higher, which would actually also fit with switch-like behavior. And the answer is no. This is actually the half-life of both S-phase and M-phase substrates, and you can see they are essentially identical and very fast. Just 2 minutes is the average, which means that actually phosphatase activity is important for ensuring switch-like behavior, but probably alterations in phosphatase activity is not playing a big role in this part of CDK regulation through the cycle. But by having rapid turnover, everything is ultra-sensitive to changes in CDK activity. This is a summary then of Matthew's work. We've identified what we think are very reliable CDK substrates. We've looked at the phosphorylation profiles. This is just to remind you up here. Lower levels of CDK activity are sufficient for S-phase substrates. Mitosis requires higher levels, and turnover is unchanged, just as I explained to you. Now, I told you we'd simplified the network by making a fusion protein and getting rid of the cyclins. But there are other regulatory modules. And the main one, which I'd spent many years studying, is, as you've heard, WEE1 and CDC25. WEE1 is an inhibitory protein kinase phosphorylating CDC2 on tyrosine 15. CDC25 is a phosphatase that takes that off. This is an essential regulatory loop of CDK regulation. There's others too, and CDK inhibitor and APC, but I just want to focus on this particular one because Damien, with his fusion protein, decided whether he could go further in simplifying this network. And to my surprise, you could. So he took this fusion protein, you remember it again, mutated the tyrosine phosphorylation site to phenylalanine and the adjacent threonine, which gets phosphorylated at a lower level, to an alanine. And this is a CDC2AF, okay, which cannot be phosphorylated on that negative site. And he also, in a similar strain, notice everything you can imagine has been deleted in this strain. Won't go list them again, but all the cyclins are out. He deleted WEE1, and there's a subsidiary WEE1-like kinase called MYC1. So that in both of these situations, you do not phosphorylate those residues. To my total astonishment, these cells are perfectly okay. Now, by this time, I was getting really nervous because I'd spent many years of my life, starting in 1975, defining all these important components, which Damien every month was throwing out of the window. It was like, you know, when you die, you're meant to see your life go backwards. So, I was watching my life's work run backwards and disappearing as everything I'd ever worked on disappeared. Okay? But these damn strains are almost perfectly normal looking. Their generation time is a bit reduced, so at least it has some effect, okay? But it isn't, frankly, that significant, and we're still investigating why. And even cell size at mitosis was pretty similar. This was particularly disturbing for me. Actually, G1 is greatly extended. That is another difference, which we are investigating, and I can't tell you why yet. But one thing that was particularly disturbing about this is that we thought from some earlier work that we'd carried out, published a couple of years before, that tyrosine phosphorylation played an important role in cell size control. You know, that is the size at which you underwent mitosis. And yet here we had almost perfectly normal cells when we deleted that control. It was quite a nice model, not right, okay? At least we showed it wasn't right, you know. This is actually cell size at mitosis. This is wild type. This is the fusion protein. This is the fusion protein, which is in the AF background. It still divides at the right size, although there is increased cell-to-cell variability in cell size. So it's not perfect. I'll just show you a couple of other experiments carried out by my graduate student, Lizzie Wood, who has just graduated. She did some time-lapse to look at the homeostatic properties of entry into mitosis. And this is shown here. Essentially, what she did is measured the sizes of cells when they enter the cell cycle, and then the size when they undergo mitosis, and asked, if you perturb cell size or exploit natural variation in cell size, what effect does it have on the subsequent cell size at division? So if you're big, do you then undergo mitosis more rapidly than normal because it's homeostatic? If you're small, does it take longer? And this you can look at with a plot here, where what you look at is the length at birth versus the length extension that occurs before you undergo mitosis. So if you're small, you have to grow more before you can undergo mitosis. If you are big, you grow less. It's a measure of the strength of homeostasis. Do you see? Good. Won't have to explain that again. And she applied that to CDC2 AF. Pretty good homeostasis. Right. So, conclusion. The AF may be influencing, that phosphorylation may be influencing cell size at mitosis. It cannot possibly be the entire story. Now, having said that, we've just about deleted the entire story as I knew it. Okay? We had run out of genes that we could turn to, despite 30 years work on this thing. So we thought we'd better, what that must mean is there's more genes out there we don't know about. Now, this has been studied by us in fission yeast, by others in other genetically tractable organisms, budding yeast, flies, in Xenopus a lot, mammalian cells, and it's all been circling around essentially the genes that were mostly discovered in yeast. What this suggested to me is we had more to learn. Now, you've heard about the Wee mutants, which are advanced in the cell cycle. This is a drawing I did in 1976, so I thought I'd just show you that. And just to show you that old papers still live on, this is a paper I published in 1975, and I just wanted to put it, as it's over 40 years old, I thought I'd just put it in here, which shows that the Wee1 mutation here, cells become advanced into mitosis. But these are the Wee mutants which are advanced into mitosis prematurely. This is my colleague at the time, Peter Fanti, he's just retired. So what we decided to do was to look for more Wee mutants to see whether we had yet more to discover that would tell us about rate-limiting steps in the cell cycle, right? Now, we're in a much better position to do that. This was done by Francisco Navarro, another Spanish postdoc, handsome fellow, as you can see here. Can't say that sort of thing in the United States because people come up and grumble about it afterwards, but I think I'm okay in Austria, am I not? Yeah. Okay, so he went for a new screen for small-sized mutants, and what he exploited was a resource that we constructed in the lab with the South Korean consortium, where we deleted every gene in fission yeast, all 5,000 genes, one at a time. So we have arrayed heterozygous deletion diploids, which have got 99% of all the genes deleted. It had been done in budding yeast some years previously, but we'd done 99%. And what this meant was, because looking for small-sized mutants is not an easy thing to do, what we could do is look for mutants which were dividing at a smaller size in each of those 5,000 deletion strains, okay? And so we actually did it with 3,000 because we wanted to work with the non-essential genes so we could actually grow them. And we went through various screens and ended up with 18 small-sized mutants which are arrayed here. This is wild type, size at mitosis. This is Wee1, which I isolated by accident in 1974, and you can see why I could get it, because it's so much smaller. What we see here is a whole range of other semi-Wee mutants, some of which we knew about before, shown up here. POM1 was one that I've studied, PIP1 is a phosphatase, but most of them we didn't know about. Now, that's amazing. Most of these we did not know about, okay? And so the genomic approach, the omic approach, has thrown up 18 gene functions, of which at least 10 are completely new to this whole field. And we haven't even quite saturated it, and this is only gene deletion, not more subtle mutations. So there is quite a lot more out there, all of this, for example, which we don't know anything about. Just in case you aren't impressed by the way of small changes, the semi-Wee, you think, are we just making this up? We aren't, and I'll just show you that here. What we've done here is made double, triple, etc. mutants of these different ones, okay? So here we have wild type. We've made a double here. This is cell size at mitosis. A triple, a quadruple, and a pentuple. We've ended up with a Wee1 phenotype. All of these are not as strong as Wee1. They are all contributing in ways we don't understand to the regulation of CDK and the onset of mitosis. I say the regulation CDK, it could be of the phosphatases, it could be of other things, but certainly the onset of mitosis. 3 of those, and remember this was a blind screen, were all part of the SWI/SNF chromatin remodeling complex. 3 of them. So it can't be unreal. It's got to be something there. I don't know what it's doing, but it shows you we have a significant resource. So Sol1, SNF5 and SNF22 are all part of that complex. So, what do we conclude from that? Well, we conclude that despite the really considerable investigation of this particular problem, that if you take an omic approach, you suddenly liberate and discover many things that we haven't found before. And this has involved many hundreds of workers on this system, and we still have genes that we don't know what they do. So, how can I sum all of this up? Well, first of all, I think what I've tried to explain to you is that the basic core principle behind the cell cycle that I think is revealed by this is perhaps simpler than what we thought was the case before. That a primitive cell cycle in fission yeast, or maybe the first eukaryote 1,500 million years ago, would be based on a increasing CDK activity that brings about phosphorylation of different substrates in the way I've described, that brings about S phase, blocks S phase, brings about mitosis. That this, I imagine, has evolved to be that bit more efficient by adding on more specialized CDKs that perhaps can be regulated more specifically, that do contribute to different parts of this cell cycle. So I'm not for one moment arguing that qualitatively different CDKs don't have roles. I'm just saying that we can understand it better by focusing on absolute activity in the cells. We know that in mouse manipulations, for example, that you can get rid of different CDKs and they can be substituted by other CDKs, completely consistent with what I've shown you, although developing a proper mouse is compromised, suggesting that part of this is probably developmentally regulated, but still the core principle stays there, but you regulate it in different sorts of ways with more complicated organisms. But it is possible to reduce the complexity of regulation and yet still get out the basic controls. Checkpoint controls, such as blocking DNA synthesis or damaging DNA, don't work in the tyrosine mutant, for example. So that doesn't work. Although there's another point to emphasize there. The cells that we're producing in the absence of that checkpoint control are very healthy. So actually those checkpoint controls are not operating very much in most cell cycles. And it just may be that because we've focused on pathologies, that we're getting this a little bit out of balance. That they are obviously revealing, but they may not have quite the emphasis that we thought. But I wanted to put it together in the last minute or two by thinking about how one should perhaps approach complex biological systems, because that's what many of us do in the room. We're faced with complex biological phenomena. Now, the first thing I think is that omics is incredibly important. Naming of the parts is essential. I thought we'd saturated it by traditional mutagenesis. We obviously have not. It's only one omics approach, but it's a very powerful one. So when today we are doing a regulatory, trying to understand a regulatory process of some importance, doing omics is important to name the genes, to know the players that we have to deal with. We then need to do the conventional interactoromics and so on to get some sense of how it operates. But then I believe we have to go a step or two further, perhaps simplifying the network to identify the core principles so that we know what is really the basis of what is going on. Because biology often has a lot of peripheral things that play roles, but maybe only in certain circumstances, and it can distract you from perhaps the core principle. And psychologically, the way we do research distracts us. If you're a PhD student and you're working on a gene, you want that gene to be interesting, you know? So psychologically, we build up what we think is interesting. Now, that's okay for your PhD, but every PhD contributes to everything being equally important, and it just may not be. It's like Animal Farm. You know, all animals are equal, but maybe they're not. And it's the ones that are not that we need to be identified with and need to identify. And we can do that through some of the sorts of manipulations that I've described, which simplify the regulatory networks, yet still maintain the basic process, and therefore get us to focus on the core principles. And that's what I've tried to do. And if I can get this to work, can you press it to get it to go? Look at this. It's the end of the sound.
So you showed that a single CDK cyclin is sufficient to drive both the mitotic and the meiotic cell cycle.
Yeah.
And in the case of meiosis, you use the trick to to have multiple copies of fusion.
Yeah.
So my question is, so I have two questions. One, are meiosis 2 substrates differentially sensitive to CDK activity than meiosis 1 and mitosis?
Yeah.
And the second, how is this related to a different threshold of APC activity in meiosis?
Right, they are both good questions, and I can't answer either of them. I haven't done the phosphoproteomics in meiosis. The first of your predictions is a good one, and I think that's what will happen. I think there will be differential sensitivity of some substrates. As we know, meiosis 1 is a reductional division, meiosis 2 a non-reductional division. I have a feeling there are substrates in there that are differentially sensitive, but we haven't done it. What was the second question again, though?
So if there is a difference, whether this is related to a different threshold of
Yeah.
APC activity in meiosis 2, then...
Yeah, so it's related. So without the first, I can't answer the second, but I think it's a good project. Doing the phosphoproteomics in meiosis is a little complicated because to do a normal meiosis, we have to starve cells, which upsets some of the way that we do the phosphoproteomics. But we can do it with a mutational approach, and it's something I would like to do, I have to say, but we haven't done it. But it's a good question. Yes.
So in the mouse, there also seems to be a grade of redundancy. For example, there are these triple cyclin mutants
Indeed.
that are still viable. So given that in the mouse or in metazoans you have also the cell cycle role maybe in differentiation, so in cell specification. So could you comment how this model that you predict could actually work there?
If you derive fibroblasts from these various lines, usually they're okay. So there isn't too much of a problem. So the basic cell cycle, I suspect, still works. But in development and differentiation, the cell cycle is intimately connected to, as you've stated, with tissue generation, organ generation, and there, I think there is more subtle regulation that indeed will work through different cyclins. So I'm sort of riding two horses at the same time. I'm saying the basic principle is you have to regulate the level of CDK activity, but to bring about complex biological outcomes, such as a mouse, you may best do that with qualitatively different CDKs, but that shouldn't blind us to the fact that the basic principle is simply increasing CDK. Is that?
Yeah.
Yeah.
Why not?
My question relates to conservation. I mean, would it be possible actually to replace in your Cyclin CDK2 fusion protein the fission yeast version with a mammalian higher eukaryote CDK and still get viable yeast cells?
I think it is possible. I have, in the last year, I've taken on 2 graduate students and suggested this as something to do. They've said no, okay, and I'm not an autocrat, so that's their choice. But I would like somebody to try it because I think it will work. And what it will do is, one problem with working with yeast is that nobody really cares too much about yeast, and so I mean, it would up the profile of what we've done if we did that experiment. But my students are more pure than me and think that's we're not here to up profile. We're there to make the discovery. I'd really like to do that because I think if we take a Cyclin B CDK1 fusion, I think it will just work like a dream. I'm sure it will. And I should just do it. Maybe I should do it myself. Now the trick is
Alex?
Probably.
So you argued that the additional variant needs to be identified. Do you think that the minimal system that you've developed would actually be the perfect background to identify those novel regulators?
It surprised me that there is so much more out there. Though do remember, it isn't having the same dramatic effects as the more extreme ones. And so I think the minimalist system is good to look at that. Because then that allows you to investigate the related problem in more complex organisms with a more sensible framework. My sort of view is, though, that once we get into the metazoa, and once we start thinking about development and differentiation, we're going to have another plethora of genes around it, which will be important for that. And unless we've got it in the simple system first, it will be difficult to sort out what's happening in the complex systems. So yes, I think it's the best way to start.
So you showed in your model that the involvement of CDKs from G1 to M phase and S phase and finally mitosis. So recently people have shown in stem cells that that these are in the sometimes they can be in the quiescence phase in the G0.
Yeah.
And the new term which is called G alert phase before they get into the G phase and back into the cell cycle.
Yeah.
So I was trying to think, what do you think? What is the involvement of CDKs in this G alert phase, and whether this G alert phase you think is conserved throughout the different organisms?
Yeah. I have perhaps a slightly unusual view compared with some of my colleagues. I don't think yeast is very useful for looking at G0 or these sorts of phases, because the point about G0 and the stem cell controls you were mentioning is that we have cells in the organism which are bathed in good nutrients and so on, and are switched off from entering the cell cycle. Yeast doesn't behave like that generally. What it does is, as long as there's nutrients there, it just goes into the cycle. And the only way you get into something like G0 is to starve it of all nitrogen or all carbon. I mean, a sledgehammer that no mammalian cell is ever abused with. So, interestingly, I think yeast is good for certain things. I don't think it's good for understanding this. So I don't think anything we've done here is directly relevant. I do think there's interesting things that are going on, but again, I'd say it's more in the territory of interacting with development and differentiation, and I don't think yeast is good for that. Not everybody agrees with me, by the way, but that's my view of it. There was a question here that's been around for some time. Yeah.
One of the obvious questions emerging from your simple cell cycle control model is what distinguishes an S-phase substrate from a mitotic substrate? What governs? Do you think the threshold at which a substrate site is phosphorylated, do you think there's any general rules that might be discovered at some point?
Well, as you can imagine, we've gone through these protein sequences bioinformatically. We looked for signatures that might show differences. We have some signatures. We are working through them. We have yet, what I'd like to do is to transfer S-ness to M-ness and M-ness to S-ness. We haven't succeeded yet, but we haven't exhausted what's there. So, will we? I'd say we're like this. Now, if it isn't a motif, I'm struggling. I'm not sure quite where to go experimentally.
Abundance, a number of potential phosphosites.
It could be, but I don't think it is, because we can look at the reports that people have made using mass spec, not us, and there doesn't seem to be much with abundance. It could be abundance combined with motif. We're going through the motive, and we might relook at abundance again in that context, I think. So at the moment, what you say is right. We are looking at it. We haven't found anything.
Well, thank you again very, very much for the wonderful talk.